So there's a lot of data in literature indicating that sulfate sources are more soluble in solution than hydroxychloride, but hydroxychloride solubilized once the pH is very low, which typically happens in the abomasum and in the duodenum. So once in the ionized form, it doesn't matter in this particular case whether the origin of the metal is a hydroxychloride or a sulfate. The mechanisms of transport for some of those metals are well characterized, such as, for example, zinc. Once it gets into the small intestine, it's illustrating here an enterocyte. The zinc, divalent zinc, can be transported by zinc transporter proteins, such as illustrated here by this protein called ZIP4. So this protein takes zinc that is in the ionic form across the epithelial, the membrane of the epithelial cell into the cytoplasm, where zinc binds to a chaperone protein to be transported across the cytoplasm. Once that zinc gets to the basolateral membrane, it's delivered to another transport protein called zinc transporter 1, where it gets into the portal drain viscera, such as mesenteric vein, and here it binds to carrier proteins such as albumin that takes zinc now through the portal circulation to the liver and eventually to the general circulation. A similar process takes place with copper. Copper has two critical transporters. One is this divalent metal transporter 1 that can carry copper 2+ across the apical membrane of the enterocyte, or copper can be reduced and gain electrons and be transformed or converted into copper +1 and now be transported by this high affinity copper transporter protein 1. Inside now of the cytoplasm, it binds chaperone proteins until it's delivered to the portal circulation, where it binds multiple proteins, primarily albumin, but also ceruloplasmin and transcuprin. This now delivers copper to the general circulation, but copper can also be recycled back to the enterocyte, and it can be released as components of bile to be secreted or excreted as part of feces. Lastly, manganese. The mechanism of manganese absorption are less clearly characterized, but there are 3 transporters in the apical membrane. The same divalent metal transporter 1 that can carry copper, can carry iron, can carry manganese, but also ZIP14 and ZIP8. These are 2 cell membrane proteins that can carry manganese. Manganese now into the cytoplasm binds to a chaperone protein that has not been clearly identified, and then it gets extruded to the portal circulation by a basolateral membrane transporter that has not been characterized, at least to the best of my knowledge. So these are general mechanisms of copper, zinc, and manganese absorption, and you can clearly see that this mechanism require transport proteins and chaperone proteins, because obviously in high concentrations those metals can be deleterious to the tissue and can be toxic. So to no surprise, many of these elements, they decrease in concentrations as calving approaches. So here's an example from work from Jesse Goff at the National Animal Disease Center in Iowa, in which he characterized concentrations of vitamins and some minerals in cows as they approach calving. So in this example here, you can see that zinc concentrations decreased as the cow approached calving and reached its nadir in the days following calving, because of output as colostrum and uptake by the pregnant uterus. And that can quickly be reestablished once that animal consumes the diet. So, but there's this loss of nutrients that typically take place around calving because of sequestration, either by the mammary gland or sequestration by other tissues, one of which would be the pregnant uterus, or perhaps other tissues in the gastrointestinal tract. So to no surprise, there's a lot of associations of mineral status or vitamin status with periparturient disease, in part because of this decrease in concentrations around calving.